Dr. Priti Mehta is currently working as a professor and head of Pharmaceutical Analysis department at the Institute of Pharmacy at Nirma University in Ahmedabad, India. Her area of expertise encompasses stability studies of drugs, impurity profiling, elucidation of degradation pathways of drugs, bioequivalence and bioavailability studies, the effects of radiation exposure on medicines, as well as the development of radioprotectors.
Before starting for the drug stability, I would like to give a small introduction from where I am. So I belong to India from Gujarat. Gujarat is the place of Mahatma Gandhi, is a known leader, and I am from the same place and belong to Ahmedabad. Ahmedabad is one of the city of Gujarat, and it is also known as Karnavati, and having a blended version of both historical places as well as the modern architecture. Recently inaugurated world largest stadium, cricket stadium in Ahmedabad, and the tallest statue was also inaugurated near to Ahmedabad. It is in Gujarat. Few monuments are also listed in UNESCO, and the way the city is having the heritage culture as well as the modern culture. I belong to the Nirma University. It is the one of the private university, and in that one of the institute is Institute of Pharmacy, along with the other institutes. And we have put our footprints in national level as well as into the state level. We are running ahead in terms of the ranking. With this brief introduction, I would like to go ahead for the talk about the drug stability. So whenever we talk about the drug stability, then it refers to the capacity of the drug substance or product to remain within the established specification of identity, strength, quality, and purity in a specified period. And always we determine shelf life of any formulation, and which depends on the type of the dosage form, types of the excipients, packaging material, as well as storage condition. So it is believed that and it is recommended by the regulatory authorities as well as pharmacopoeias that any formulation should remain in its original quality up to its shelf life, and around 90% of the drug should retain in majority of the cases. Now, when we talk about the drug stability, then drug stability is classified into physical stability as well as chemical stability. So physical stability, which is related to solubility, appearance, measurement of the rate of dissolution in solid dosage form, drug release study, etc. And when we talk about chemical stability, then all other chemical reactions which can happen to the drug during the shelf life is measured, which also include drug excipient compatibility studies as well as drug-drug interaction in combined dosage forms. If we talk about the drug stability evaluation parameters, then normally it depends on the dosage form. So in majority of the dosage form, like solid dosage forms, semi-solid dosage form, liquid dosage form, the physical parameter evaluation get changed, but for the chemical parameter evaluation, the major thing remains as assay determination as well as impurity determination. So the assay and impurities, information about the drug present in the formulation as well as the different types of the degradation products or impurities present in the formulation give us an idea about chemical stability of the particular drug in a particular dosage form. When we talk about the stability evaluation of any drug, then degradation behavior of the drug is measured. Now, if we would like to determine the degradation of the drug in API, that means in active pharmaceutical ingredient, then that degradation will follow simple reactions. And by calculating the degradation rate, using Arrhenius equation or other kinetic rate equations, we can easily predict the degradation behavior of the API because it is a only sole component in API. But when we talk about degradation of drug product, then it is a complex process compared to API degradation, because here matrix effect also play important role. So here the active ingredient or drug is present in atmosphere of the excipients, like a solid formulation have a different excipients, colloidal suspension if it is there, then having a different excipients, likewise emulsion, gel, palantrals, etc. So the impact of this excipients on the stability evaluation of the drug is important when drug is in formulation. The evaluation of stability is very important because it helps us to give us an idea about the maintenance of it, the quality, efficacy, as well as safety of the patients. Quality, that drug component or the drug concentration is within the specification. Efficacy, that means that appropriate amount of drug is present and so it can give desired action. And safety, the degradation products are within the control, and that's why it is not leading any toxic effect into the patients. So which are the different parameters affecting the drug stability? We are talking about the drug formulation. So the inherent property of the drug itself, physicochemical property of active ingredient and excipients, which can lead to the drug stability. So few drugs chemically they are very stable, few drugs chemically they unstable, and that's why it is an inherent property, chemical property of that molecule. Second, composition of the dosage form and manufacturing process. We know that paracetamol tablet is much more stable compared to paracetamol injection or acetaminophen injection, or any drug if it is present in the solid dosage form will give more stability compared to the drug present in the liquid or semi-liquid formulations. Third, which type of container closure system is used for the packaging material, which protect the drug from the environmental condition of the, and last one is earth environmental factor. So first three factors, the first factor is the inherent property of the API or drug, which we cannot alter. Another two factors we can optimize by changing our manufacturing process or by changing our packaging material, but we don't have any control on earth environmental factor. The drug stability is evaluated by taking few environmental earth factors in consideration. So drug is exposed to higher temperature, higher humidity, or light conditions to check its stability. And this is known as accelerated stability studies. And these studies are used to predict the shelf life of the drug. And these studies are carried out not at the storage condition of the drug, but it is carried out at higher conditions, higher temperature, higher humidity, and light exposure, and which give us an idea about the stability of the drug. We can extrapolate the results of the accelerated stability studies to the shelf life of the dosage form. So the shelf life of the dosage form can be predicted within the months by doing the accelerated stability studies. This practice we are doing on Earth to check whether drug is in stable form or not on Earth. To do such studies, ICH guidelines are available. So International Conference on Harmonization Guidelines, Q1A to F, all are related to do the stability testing of the drug, either in the accelerated condition or into the real-time stability studies. And Q1A is the gold standard to conduct the stability studies. Along with that, there is a specific guideline, Q1B, which talks about photostability testing of new drug substance and products. If we talk about these guidelines, they are based on the earth environmental factor, which we have discussed earlier, temperature, humidity, and light. Now we would like to send our medicines to space along with the astronauts. And we know that space exploration is increasing. The space agencies are talking about long duration space missions like Mars mission or lunar mission. International Space Station is functioning since many years, and astronauts now staying on the station for longer duration in months, and few companies like SpaceX, they have, and other companies also, they are talking about the space tourism, and they have invested money for that also. So they would like to send people for the space tourism. So as the space exploration is increasing, more people will explore the space in future. And as the people will explore the space, medicine will be important parameter to be used by the astronauts during space exploration. For any human space exploration, the preservance of the health and wellness of astronaut is very, very essential. And for that, efficacious pharmaceuticals with adequate shelf-life is also essential. It is also important to maintain the stability of the drugs during the space mission. We know that space mission can be of long duration, it can be of short duration. So for any of the space mission, it is important to check whether the pharmaceuticals are stable or not. If we see about the use of the medical system from Mercury mission to right now ISS, Dr. Virginia also mentioned it very nicely that previously it was hardly few medicines. Mercury mission started with 3 medicines, and now it is increased up to more than 120 medicines to be used at ISS. And it is keep on adding as per the requirement, as per the clinical symptoms observed in the astronauts. And in ISS, right now the crew health care system is divided into countermeasure systems, environmental health system, and health maintenance system. And health maintenance system include medicines, first aid kits, and other life support system. If we look at few reported articles, majority of reports are available from NASA. I have used the data from the reported reports available, and I have mentioned it in my bibliography references all together. But this all are the reported data which I am showing over here. So it is observed that in all the space flight records, in majority of the cases, medication was used by the astronauts, and it is also reported that the effect of the drug was less than the anticipated, or either medications did not work at all. So as Dr. Virginia told that after having a dose tracking system, they tracked that instead of few medicines, now the dose of medicines taken by the astronauts are also much more. So, it shows that the astronauts are taking medicines also repeatedly, but they are not able to get the effect as they are assuming or as it is anticipated. That means particular dose is taken by the astronaut to treat the headache, but it did not relieve the headache completely, or they have to take the another medicine likewise. What can be the probable reason for that? One reason we have discussed is human space physiology. Yes. One of the reasons that due to the fluid shifting, due to the change in the drug distribution, due to the change in the metabolic profile, there are chances that the desired drug effect is not observed. But the another factor we cannot overrule is the space environmental factor. What are the reports available regarding the stability of pharmaceuticals in ISS? Many reports are available. I have taken here 2 examples. One related to the quality of the medicine stored in ISS. So the drugs were stored up to 28 months in ISS and then evaluated compared to a ground control. And it was found that only 9 ISS stored formulations met USP acceptance criteria. But compared to that, 17 ground controls met the USP acceptance criteria for potency. That means assay. So here you can see the drastic difference in the number of formulations shown inability to meet the assay requirement or potency requirement. And it is also observed that phospholipid drugs like promethazine, dextroamphetamine, ciprofloxacin, they degrade in higher rate, or the percentage degradation is more in space compared to ground. Another study reported is to evaluate the stability of vitamin B complex in 2 brands, which were stored at ISS for 4 months. Same vitamin B complex, but manufactured by 2 different companies, and it was stored. So it was found that one company, the one brand, was falling outside the regulatory requirement. That means it is not meeting the potency criteria as per the pharmacopoeia. But in other formulation, it is meeting all the criteria. That means the excipients also play important role in stability of pharmaceuticals. This figure I have directly taken from the report published in 2019, and it was so many studies are carried out by different group of people, and Dr. Watting also have published very good articles regarding the drug stability. And you can see that here all the red color medicines are not meeting physical or chemical stability once it is evaluated after returning from ISS. In all the studies, you can see that few drugs or many drugs are not meeting the criteria of potency once it get retrieved from the ISS and evaluated for its chemical and physical stability on Earth. This is due to what? What are the factors responsible for it? To know about it, it is important to know about the different environmental factors of the space. Space environment is, when we talk about space environment in space shuttle, then, or space shuttle or space station, then it can experience excessive vibration. Also having a varying temperature and humidity. They have to experience microgravity as well as radiation. If we talk about other 3 factors like excessive vibration, at that time there are chances of having a physical instability into the formulation. Then another is varying temperature and humidity. In the space, temperature and humidity is not controlled as it is on Earth. But we have to store medicine in space shuttle where temperature and humidity is very strictly controlled. Temperature is maintained between 21 to 30 degrees centigrade and humidity is also maintained. So it is not the factor which can affect the stability of medicine. Another is microgravity or zero gravity, which have a huge impact on human physiology. But if we talk about the medicine, then microgravity don't have much effect or not that much effect on the stability of the medicine. And many a case it happens that the formulation remains stable. Physical stability of the formulation is more because there is a lack of gravity and drug cannot get separated, or phase separation is not visible in semi-solid or liquid, semi-liquid formulations. Even in suspension, sedimentation is not observed. Physical stability is retained. Another factor is radiation. Now we have to evaluate whether this radiation is having any effect on the stability of medicine or not. Now, to know about the radiation impact on the stability of medicine, it is important to know about the difference in the radiation environment on Earth as well as in space. Space prevailed with ionizing and non-ionizing radiations ranging from radio waves to X-rays and particulate radiations. Out of that, magnetospheres as well as atmosphere act as a protective cocoon and protect us from all diverse radiations. So only visible radiation as well as somewhat ultraviolet radiations and radio waves can penetrate Earth atmosphere, and we can experience that radiation only. Outside the protective cocoon of the Earth, space is brimming with the different types of radiation. Mainly, it is divided into 3 particles trapped by the Earth's magnetic field, which include all the electrons, protons, some heavy ions. Galactic cosmetic rays consist mainly of protons, 85%, and other alpha particles, and solar cosmic rays. The composition of the space radiation is high, is I have mentioned here, and which include gamma rays, radio waves, electrons, high energy particles, and few electrons, protons, alpha particles also. So this is the composition of the space radiation environment. Check the impact of the radiation on the stability of medicine. All this radiation or all such type of ionizing and non-ionizing radiations are not much important to us because medicines are stored inside the space shuttle. And we know that the space shuttle is also constructed in a way that it can allow or it can reduce the radiation penetration inside because astronauts are living inside. So all the non-ionizing and ionizing radiation cannot penetrate the space shuttle, but there are chances that due to the interaction of the particle ions, particulate radiations, as well as the high energetic particles with the material which is used to construct the space shuttle, they can generate secondary particles. And these secondary particles will produce complex radiation environment inside the space shuttle. The knowledge about such radiation, which is inside the space shuttle, is very, very important. Which different types of the secondary particles are, generated. Normally, the neutrons, then recoil protons, heavy ions, gamma radiations, X-rays are the secondary particles generated due to the interaction of the primary space radiation with the material of the space shuttle. And we don't know the interaction of such radiation with the medicine. It is the dosimeter is used to measure the radiation dose inside the space shuttle, and it is observed that it is very well documented that though the shielding is carried out from the radiation, but still the total radiation dose is much higher, or it is higher in the space shuttle compared to the Earth. So astronauts are exposed to the higher radiation dose compared to the Earth, as well as if medicines are stored, they are also experience higher exposure to the radiations. Now, when we evaluate the stability of the space medicine, which is flown to space, then this stability is evaluated on Earth based on the stability guidelines given by the regulatory authorities, and these guidelines are based on the environmental condition of the Earth. Normally, right now, what the agencies are doing, I am taking a reference of the International Space Station, that in the International Space Station, medicines are replaced every 6 months. Or they are replaced before the 6 months of their expiry to give the quality medicines to the astronauts. But it is not feasible when we are talking about the long duration space mission or the lunar mission or Mars mission. In the regulatory requirement given in ICH, out of all the guidelines, now we will focus on the photostability guideline because it is talking about the radiation. So if we look at the Q1B guideline, which is for the photostability testing of new drug substance and products, as per this guideline, they have advised to use 2 light sources. One light source which can produce the output at D65 and ID65 emission standards. That means D65 is internationally recognized standard for outdoor daylight, and ID65 is equivalent indoor indirect daylight standard. So these 2 standards one has to use, and to produce such type of radiation, they can use different light sources like xenon, metal halide lamp, etc. Another option is to use cool white fluorescent light, which can mimic the radiation of indoor daylight, and ultraviolet lamp, which can emit the radiation between 320 to 400 nanometer. So these are the options suggested. Now, when you evaluate the drug, evaluate the stability of drug using this light sources. That means you are evaluating the stability of the drug in presence of UV radiation as well as ultraviolet radiation. In market, such type of stability chambers are available, and it is known as photostability chamber. So where both the light sources are placed, you have to put the medicine inside the chamber, and you have to store the drugs for 14 days into such stability chamber. So the drug will get the light exposure, and if drug is photolabile, Then it can show the decomposition or degradation and degradation products on evaluating using analytical methodology. But this guideline cannot address the stability of drug under different radiation conditions other than UV and visible radiation. Normally, drugs are directly exposed to this radiations. Then it is exposed into its immediate pack, and it is also exposed to the marketed pack, so secondary packaging material. As per this ICH guideline, you have to expose the drug directly into its immediate pack as well as into the market pack, and based on that you have to show whether drug is stable or not. Now, when the drug is interacting with radiation, it can be UV visible radiation. Then if the drug is photolabile, then it can show the photochemical degradation, and which can lead to the discoloration or the color development or cloudy appearance of the product or loss of viscosity or precipitation of active principles, alteration in the dissolution rate. All this type of the physical changes you can observe, as well as you can observe the loss of potency, and which can lead to the inferior drug. When we talk about the ionizing radiation interaction with the drug, then it is well understood by the radiation quality. That means it is known that different type of radiation cause different types and the level of damage to the medicine or to the material. So it is the inherent property of the radiation, and this radiation quality depends on the linear energy transfer, and it can be distinguished by the energy deposition pattern, which is reality for the medicine also. So radiation induced damage can be classified into direct ionization by direct ionization and indirect ionization, where the radiations are directly interacting with the drug, and in indirect ionization the other matrix is interacting with the radiation, and finally it is giving the impact on the stability of the medicine. Now, to show this impact, we have tried to evaluate the amlodipine bacillate to expose it with the different types of radiation. So here API and formulation is exposed to ionizing radiation at different doses and evaluated physical and chemical changes by the analytical methods and compared its degradation profile. So different types of the proton, thermal, neutron, fast neutron, gamma radiations are used as per the facility available with us, though the energy as well as the dose is not as per the space medicine, but we try to evaluate it. So if you see the HPLC chromatogram, you can see that when the drug is exposed to photodegradation, then it generates a different degradation or the impurities. And when it is exposed to the gamma radiation and proton, then different degradation products are generated. And this is due to the, we can predict that it is due to the different types of radiation involved in the interaction of the medicine. So here with the gamma radiation, so many different degradation product is observed, which is not with the photodegradation, and with the proton, another degradation product is observed, which is not with the gamma radiation or photoradiation. And we try to evaluate the probable degradation kinetic and the generation of the degradation profile. This we have carried out on Earth, but because it is not feasible every time to send the medicines on the space and to evaluate it. But these are the approaches. Right now, we don't have that type of approaches to assure the stability of space medicine. But this can the approaches we can use to evaluate the pharmaceutical stability. One is to use the space hardy formulation. Another is to use the proper storage condition, and third one is to use the ground-based simulation models or the prediction models. When we talk about the storage and packaging, on Earth we are using amber color foil or aluminium foil to protect the drug from the UV visible radiation, which is not useful to protect the drug into the space. We can use low molecular weight, atomic weight material, high density polyethylene material with the fillers to protect the medicine. So such type of packaging material can be developed. Ample of opportunities available to do research in this area. as well as to store the drug at cryogenic temperature. That means at very lower temperature, which can preserve the drug at very lower temperature. And that can be one of the options. Another approach is to develop the space hardy formulation, where we can use the excipients, or we can select the excipients. For an example, the starch, stearate, cellulose, dextrose, if they are used as excipient, they can give more stable formulation, because it is proved by during the gamma sterilization methodology that such type of excipients give stability to the formulation. And other excipients like alumina, silica, polyethylene glycol, they degrade the drug in the presence of radiation, ionizing radiation. Another approach is to use antioxidants, which can quench the oxidation species and can reduce the oxidation of the drug and protect the drug from the instability. Another approach is to use the novel drug delivery systems like nanotechnology or microtechnology. It is reported that such type of technology can help to reduce the radiation effect on the stability of the drug, but it to be evaluated for the space medicine. Another approach is to develop the ground-based spaceflight analog. There are some problems because right now such type of analog system is not available. So such type of analog system is to be constructed. Likewise, we have a stability chamber. We can have some chambers or some type of the device where we can expose the medicine and we can get an idea how different types of the radiation impact the stability of the medicine. Another futuristic approach is to continuously monitor the drug stability by using the non-invasive methodology like Raman spectroscopy or near IR spectroscopy. We can place such type of probe with the medicine and they can give us the real-time data on the ground regarding the stability of the medicine. But that type of methods to be used or to be developed with chemometric methods or with some statistical regression tools. I have seen one area in which the talk is scheduled regarding the 3D printing technology or the pharmaceutical synthesis, which can be carried out on board in the space shuttle before using the medicine. So whenever medicine is required at that time, only the medicine is prepared. So the stability issues can be overcome. I would like to show the published article in this area. One is published in European Journal of Pharmaceutical Sciences and in Journal of Pharmaceutical and Biomedical Analysis. And one book chapter also we have published in Physicochemical Stability of Space Medicine in Handbook of Space Pharmaceuticals. So to summarize, I would like to say that the take-home message is different ionizing radiation may have a different effect on the stability of the same drug and the dosage form. So it is important to evaluate the impact of the ionizing radiation on the same drugs. Second, if the drug molecules are different, then also the same radiation can give different degradation behavior. It is required to be evaluated. And hence, it is a need to investigate the impact of the space radiation on stability of the drugs flown to space. These are the references I have used for my presentation. I acknowledge the people who have worked with me to do the studies. Thank you very much for the time and thanks for providing this platform.